everyone to another episode of Nerd RX podcast, and I am your host Barkha. Well, today is a very special episode because this is the very first episode of a new series that will be a monthly special. It will come once a month where I talk to a PI and talk about their research and what are the hurdles they face in their research and what are their thoughts on solving those issues. And my one question to them is, what is one question that you would like to get answered in your lifetime? So for the very first episode, we have Dr. Aurijit Sarkar, and we will discuss the issue of antimicrobial drug resistance. Welcome, Dr. Sarkar, to the show.
Thank you so much for having me.
Thank you so much for giving us your time. And before we talk about the issue of drug resistance, we would love to hear about your journey and how did you get to this point in your career?
It's rather a long story. I think I knew that I wanted to do science when I was in middle school. Um, at the end of my high school, I remember my family telling me, "You need to get into engineering." And I said,
Uh-huh.
"Heck no, that's not going to happen." I knew that chemistry is where my interests lay. I got into a bachelor's degree where I focused on pharmaceutical chemistry, chemistry, and zoology. Then I got a master's in applied chemistry, specializing in fine chemicals and drugs. Went ahead, taught for a couple of years at the university level. This is early in the 2000s.
Hmm.
Then I got into a PhD program at VCU,
Hmm.
where 2005 to 2010, I studied for my doctoral degree.
Hmm.
Went ahead to get a postdoc at the University of Dundee, um, in Scotland, then moved back to the US for my second postdoc, which is approximately when you and I first met.
Yeah.
And then after that, after my second postdoc, I had this opportunity here at High Point University to work on building a new school from scratch.
Hmm.
So I'm currently founding faculty and assistant professor in the Basic Pharmaceutical Sciences Department at High Point University School of Pharmacy. That, in a nutshell, is my
Hmm.
background.
Okay. Wow. I think even I did my master's in Scotland.
Who
So
did you?
it's like
Where?
you.
Glasgow?
Yeah, Glasgow, University of
Hmm.
Strathclyde.
Yeah.
So yeah, that was a different experience. Like when you see two different countries, you see how different things work
Oh,
everywhere.
yes. It was a fun time. We
Hmm.
were actually in a hotspot, honestly, in Scotland because Edinburgh, St. Andrews, Dundee, even Glasgow to a certain extent were so close to one another, and all of
Yeah,
them were top institutions.
yeah.
And that's actually where my interest in serious drug discovery against microbes actually peaked. It started a long time ago, but it peaked in University of Dundee. Um,
Hmm.
we were part of this program called Aeropath, and it comes from aero as in Pseudomonas aeruginosa and
Hmm.
pathogen, so Aeropath. And they had a EU grant for targeting Pseudomonas, and it was a lot of fun. We had a ton of synthetic chemists, a bunch of crystallographers, biologists. Our collaboration was between Dundee, um, St. Andrews, Karolinska Institute, and a couple of pharmaceutical companies. Um, we actually tested out drug discovery via high throughput screening and design against a number of Pseudomonas proteins. It was an
Hmm.
amazing experience. That place is crazy in terms of the
Hmm.
quality of science that they do.
Wow. Well, okay. So my one and only question to you is, what is that one question you would like to get answered in your lifetime?
I think that plays back into what it is that I wanted to do for a living, right? I really
Correct.
wanted to study antimicrobial drug discovery. That was definitely my goal
Hmm.
from when I was very young. And in all honesty, a lot of people work on it.
Hmm.
But there are some significant hurdles that need to be taken care of before we will be successful in that area. Um,
Hmm.
so I think the one question I really want should be answered by the time my career is over is how do we bring back the heady days of the 1960s and 1970s when infectious disease was really not a problem.
Hmm.
At least in our minds, it wasn't, right?
Yep. Yeah.
Clearly, we didn't know what we were up against. So,
Hmm.
I'll give you a bit of a background story into this.
Yes.
Um, so, in the 1990s, even the early 2000s, this is when our scientists had basically mastered recombinant DNA technology. They were able to express any protein that they desired and study its qualities, um, study its inhibition, identify drugs against it. We had libraries with millions of chemicals across multiple pharmaceutical companies, which were honestly uh, not entirely redundant. But across the last 30 or 40 years, we've basically come up with maybe a handful of medications. That's it,
Right.
with novel modes of action. We haven't been able to
Hmm.
identify new antimicrobials. And at the same time, we hear of superbugs, uh,
Correct.
basically resistant pathogens which are coming up and are able to um, survive therapy using antimicrobials. They just live on, no problems there. So that's what we need to fight against. And if we're able to actually go back and figure out how we can bring back novel antimicrobials or bring back the ability to use our older antimicrobials, I think we will be in a much better position than we are at this present moment. So I think that's
Hmm.
my goal for life. That's if I can answer the question how we can do that, that I think will satisfy me.
Hmm. Okay. And why do you think we landed in this position in the first place? Is it overuse of antibiotics or just the microbes being more like mutating genetically? What do you think that is?
Both. The answer is
Okay.
both. So once we came up with antimicrobials, we thought, we're the chosen ones. We are untouchable. We started to use antimicrobials for everything. So some of the biggest problems that we see, um, for example, is the use of azithromycin, regardless of whether or not a person has bacterial infections. So children, especially, are extremely susceptible to viral infections. They pick it up at daycare. They pick it up at school. Parents are impatient and they expect the doctors to give out antimicrobials. And guess what?
Hmm.
Azithromycin is given out like crazy, and which leads to resistant infections, honestly. As patients, we don't really understand how to use antimicrobials. We do not complete the therapeutic regimen that we're put on. So, if we do have
Correct.
a bacterial infection, um, and we haven't completed our course of antibiotics, we end up um, giving rise to resistance. So that's one angle, right?
Hmm.
The other thing that happens is we've started using things like chloramphenicol and beta lactams in agriculture. So we want to make sure that our meat supplies last for longer, that they're not hampered because of bacterial infections. So we preemptively use antimicrobials in things like pig feed. tons of antimicrobials, which are diluted out, move into water supply, into the soil, exposing bacteria to dilute concentrations of antimicrobials, which then have the opportunity to gain resistance against them.
Mhm.
So, both medical use as well as agricultural use, those are two uses that come to mind right now where inappropriate use of antimicrobials has given rise to resistance. The other thing that has happened is, and this has been apparent to scientists for a good long time,
Mhm.
nature has been kind enough to provide us with natural antimicrobials, and that is how most of our antimicrobials have been identified from natural sources, which honestly means fungi. We use
Mhm.
fungal metabolites as medications. But we have to go back to the question, why do fungi produce antibacterials? Well, that's because fungi and bacteria have been fighting this open warfare for eons. I
Mhm.
mean, mankind has been around for, I don't know, a few thousand years. Bacteria and
Mhm.
fungi have been fighting their chemical wars for, I don't know, millennia. So they've
Mhm.
actually evolved chemical weapons against one another. They have also evolved ways in which they can defend themselves against those chemical weapons. So here's a really cool example. There was this one paper, and I've forgotten the name of the authors, but if I remember correctly, they were from McGill University in Canada. Um, they went to the I think it was close to the North Pole somewhere, where they dug deep into the permafrost and took samples of bacteria that were essentially buried under ice for millions of years.
Right.
They grew them out and they
Mhm.
genetically tested those samples to see what sort of antimicrobial um, resistance genes they could identify. And guess
Mhm.
what? They actually found genes for resistance against most of the antimicrobials um, that exist. These are
Wow.
bugs that have never been exposed to human antimicrobials.
Yeah.
These have been buried under the snow for millennia, well before humans came onto this earth, right? How
Correct.
did that happen? Well,
Yeah.
it's basically because of what I just told you. Bacteria and fungi have been fighting chemical warfare for a long time, and
Mhm.
there are um, genes, resistance genes, already present in bacteria. So all they need to do is enhance those genes or transfer those genes to one another, and they can continue fighting against us as well as the fungi.
Mhm. Mhm.
So, to answer your question, it's not just the overuse or inappropriate use that humans have succumbed to,
Mhm.
but also the fact that bugs have an immense capability to resist antimicrobials. And they've had these abilities for a very long time. We're just finding out about all of this in the last
Right.
few years or so.
Wow. So they did not even need to be exposed to humans to possess those genes.
Mm,
They were already present.
absolutely. I mean, that was when I read that paper, I just, my mind blew. I just said, oh my
Wow.
gosh, I can't believe this.
Yeah. Absolutely. Wow. I honestly believe, how people talk about how the world is going to end, wars and nuclear weapons. I honestly believe it's going to be one of the bugs that is going to, like, we just saw what COVID did.
Yep.
So I honestly believe it's going to be either a virus or a bacteria. What are your thoughts on it?
Don't get me started on the T virus theory, right? I think I think a lot of us have been worried about pathogens and what they can do to us for a good long time. I'm fairly certain that there's some truth to it. That there's a risk that um, we have overlooked for decades. think about what happened in the medieval period, right?
Mm-hmm.
Across about 100 years or so, maybe something like that. Basically, several decades. About half of Europe's population was wiped out by the bubonic plague.
Right.
Now, it didn't happen at the same time. It happened slowly and steadily and across a good period of time. Uh, but it's amazing that one bug, one pathogen could do this to us.
Yeah.
Look at the number of pathogens that surround us.
Mm-hmm.
We're hearing about um, cases of pathogenic amoeba getting into the brain just because somebody went swimming into a lake.
Yeah. Mm-hmm.
We hear about, um, I think I can't remember, but I think it was polio, wasn't it? There was at least one
Yeah.
case recently recorded.
Recently, yes.
So
Mm-hmm.
that's coming back. Um, Tuberculosis never left. We
Yeah.
managed to control it really well, even in the third world countries, but it never
Mm-hmm.
really left.
Yeah.
Smallpox. I wouldn't be shocked if smallpox came back.
Oh, please.
We called it eradicated, but it still exists.
Yeah. Even polio was kind of eradicated, but now you see in New York, out of all the
Right,
places.
precisely. So I don't think that it will be one particular disease. I mean, the probability of that happening is going to be pretty low. But if mankind
Mm-hmm.
is to be wiped out, it'll probably be multiple diseases because of resistance to
Mm-hmm.
begin with. But in all honesty, I feel that that's a really low probability situation.
Mm-hmm.
I think we're going to find a happy balance between
Mm-hmm.
infections and human life continuing.
Mm-hmm.
Just like we have trouble completely eliminating pathogens, they'll have trouble completely eliminating humans. I don't think we're going to go
Wow.
away. But yeah,
Yeah.
I mean, it's still going to cause immense damage. That's nothing to sneeze at.
Right. Wow. So, in your opinion, how do we tackle this issue?
People have been trying to answer that for the past 50 years and have failed.
Yeah.
I think we're going about it the right way to some extent. Clearly, there are problems, but we have to find ways in which we can increase our productivity when it comes to fighting resistance.
Hmm.
So it's not just the science that is stopping us from being successful. We also have to find some power in terms of financial commitment. So
Right.
a big success that we are seeing at this time is in the clinic, where we reserve the newest antimicrobials for the worst of infections. Clearly, right? We think that
Mm-hmm.
because these new antimicrobials have never been used, then resistant infections are unlikely to happen, in which case those antimicrobials are actually present to help when needed. True,
Yeah.
very true. But you know what that has done? What that has done is, in the current capitalist um, approach towards um, fighting antimicrobials, there's no incentive for companies to come up with new medications because they won't be used.
Mm-hmm.
If they're not used, then the companies can't actually even break even with their investments. So it
Mm-hmm.
makes zero sense for them to invest more money in antimicrobial development. And this has been a problem since the late 1990s, and we still haven't overcome it. We need
Mm-hmm.
to find ways in which they will be paid in terms of the value that the medication brings to society rather than just simply the actual sale of the product itself. There are a number of other approaches that much more learned people than I have proposed. I won't get into each and every nitty gritty detail, but I'll just keep it at the bird's eye view level. Is bird's eye view level that um, unless we are able to solve the financial conundrum, which is to incentivize the use of or the research on a product that will not be sold, it might be a bit difficult to fight resistance. And I hope I said that right. That was a really complicated sentence.
So there are so many things we already have issues with. Like we recently saw the COVID pandemic. We also now have the drug resistance issue and the opioid pandemic. How many more things should we worry about?
And they're all interconnected, you know. So let me give you an instance. So there was a study by Fleischauer et al. In MMWR, excuse me, which did two cross-sectional studies, one in 2010 and the next one in 2015, on infective endocarditis in the state of North Carolina. And guess what they found? Across that 5 year period, the instances of infective endocarditis had gone up 20-fold.
Wow.
Now, infectious endocarditis basically is it's caused by a number of different pathogens, but the most serious one is Staphylococcus aureus. So MRSA being one of the
Mhm,
biggest ones. Sometimes Visa, although they're still relatively rare. So what they found now, clearly the type of study that they had done is not meant to show causation,
mhm,
but across that same 5 year period, there was also a huge increase in the use of illicit drugs, particularly heroin, which are taken IV,
mhm.
and people were sharing dirty needles,
Yeah.
which very often spreads things like staph.
Mhm,
So not only are we seeing these individual problems like the opioid epidemic and resistance, we're also seeing a marriage between these different problems where one is leading to the other. And that is a huge problem in North Carolina because we're at the crossing of 95 with 85. It's a
mhm,
rather busy corridor. So clearly, it's one of the reasons why drugs are spread in this area bunch.
mhm.
But this state is not the only one that's seeing the same issue. So there have been similar reports from, I believe, West Virginia and Virginia, as well as certain other states. Everybody's seeing the same thing. As people use dirty needles in order to take heroin, they're also spreading infectious agents and resistance. It's
That is
crazy.
one marriage I do not want to attend.
No, definitely not. Me neither. I'd much rather go away somewhere else.
What are your thoughts on drug repurposing? Like I've seen now, because you know how expensive is developing a whole new molecule, and most of the time, I would say like more than 90% of the time, it fails. So now we have seen a surge of older drugs being used to treat something new. So, do you think drug repurposing is something that would help fight this problem?
It's a good stopgap measure.
Hmm.
So I'll give you one example. Um, a colleague of mine at High Point University, her name is Megan Blackledge, wonderful, wonderful researcher and teacher. She actually ran a really nice project along with Patrick Viguera, also from High Point University, up until recently. Um, they came up with a couple of classes of drugs. Um, there was loratadine, and then there was another one. It was a tricyclic antidepressant, and I can't remember which one it was. But both of them, at roughly about 50 micromolar, would actually enhance MRSA to oxacillin. So that's amazing. That's really brilliant.
Hmm.
The only problem is that The medication itself, which is FDA approved, we need roughly about 1,000-fold more than we would typically use in a patient to achieve that same outcome.
Okay.
Uh, just to clarify, so
Mhm.
in order to enhance oxacillin against MRSA, you would use roughly about 10, like 50 micromolar, excuse me. But I don't think you can use that higher concentration in patients, as has been
Mhm.
evidenced by clinical trials across
Mhm.
decades. So it's a good starting point, and there will be some situations in which an FDA approved drug can automatically be shunted into um, the antimicrobials arena.
Mhm.
But I think it's a good source for initial hits rather than the final product, in which case you still have to go through the whole development cycle, phase one, phase two, phase three, then approval. It'll be cheaper, yes, because you don't have well, you have mastered the synthesis essentially. You can produce it by the
Mhm.
ton, but it won't be ideal because remember, again, the product is changing.
Yeah.
So we're still left with the same problem of um, going through approvals for a new drug, which we were hoping to avoid, if that makes sense.
Yeah.
So it's a good
Okay.
stopgap measure. I think it's great for initial hits. But
Mhm.
I think a good approach towards that would be to begin with the clinical endpoint itself. So clinicians will have to go through medical records and perhaps run things like cohort studies to identify whether or not certain medications actually reduce the risk of infections. And that sort of data is really difficult to find because, for example, I mean, who would look to note whether or not somebody who's taking loratadine or a tricyclic antidepressant actually ended up getting an infection or not?
Mhm.
Or if you did it in the other way around, so if you did a case control study where people have infections and do not have infections, and then you're looking to see if you took loratadine in the past or not, hoping that the loratadine usage in the uh, uninfected group is more. Those are really difficult studies to perform, honestly. But it might be one way of identifying immediately viable chemicals. Chances of success are really low.
Okay. So I think starting with the antimicrobial, like the typical path, like computational, and then SAR studies are more, you would say, more fruitful.
No, that's the other problem. So
Okay.
that's precisely what we were talking about. Happened in the 1990s and the early 2000s is we thought, well, we can produce the protein, we can do computation, we can design medications. Medicinal chemistry pathways are pretty well established, and it's a mature art. We ought to be
Right.
able to identify new drugs against microbes like that really quickly.
Yeah.
Yeah, no, never happened. And the major reason I think was because we were not looking at the right places.
Hmm.
So the bacterial outer layers are extremely different compared to the human outer layers, cellular outer layers. So what happens is all of the pharmaceutical companies like to synthesize chemicals that will get inside humans. Um,
Right.
so if you pop a pill, Lipinski's rule of five compliant, chances are it's going to get through. Um, there are problems there too, but I'll tell you what, that's okay. Those chemicals are known to spread through the human body.
Hmm.
The problem is, especially in gram negatives, which are the more worrying of infections, emerging infections, these chemicals either just simply do not get inside the bug, or else, if they do get inside the bug, there are efflux pumps present which are kicking the chemical out.
Mhm.
So I think part of the problem is now being solved. The Gurskaya group in Oklahoma has done some excellent work um, in understanding chemicals, the rate at which chemicals will be effluxed. There's a group in University of Illinois at Urbana-Champaign, led by Paul Hergenrother, who have done a brilliant job of understanding chemicals which will get inside the bug through diffusing through porins. Uh, Fantastic work, honestly. I think those sorts of things need to be done more. Each bug needs to be addressed differently. So if you look at the escape pathogens, um, you have Klebsiella, you have Pseudomonas, you have an Enterobacter. Um, each of these has a separate arsenal of efflux pumps, and all of their outer layers are somewhat different from one another. Porins have a different character. So we need to understand the effect of all of these things in a mechanistic manner to identify which chemicals will get inside the bug. Once
Mhm.
you understand the properties of chemicals which accumulate inside these bugs, then that's the chemical space within which you have to identify novel antimicrobials. And
Mhm.
I think all of that is being done.
Mhm.
And I can already see that at least one generation of researchers who are now very senior researchers, very well renowned, are winding up their projects. But it is not done yet. We'll need
Mhm.
at least another couple of generations of researchers whom they are training, who will take up the mantle and end up doing something, uh, the sum total of which may help us understand which chemicals we need to look at. Then there are
Mhm.
people like me who have worked on drugability. Some of my most highly cited papers are related to drugability of proteins. In other words, if you have this particular pocket in a protein, will it bind a drug like chemical or not? But that has to do with chemicals which are getting inside the human body, not inside
Right.
the bug. So we have to rethink those drugability issues next.
Mhm.
Then, after that, you have to find novel antimicrobials, and then perhaps we'll be safe. So I'm expecting that by the time my career is over, we'll be getting closer. But who knows?
Wow. Just closer, not solve the issue.
Right, exactly.
Wow. Okay. And I think I would like to just ask, what is one advice you would like to give researchers like new researchers like myself in who are in the antimicrobial research?
Not just you, me too. In
Yeah.
all honesty, I mean, yes, I've been an assistant professor for about 6 years, or going on 7 years now, but even I'm still learning.
Hmm.
So, one word of advice is never stop learning,
Hmm.
because, um, in all honesty, when we start reading other people's research and we have a specific interest that we try to figure things out on, even if it is not in our current arsenal of expertise, we start learning things that we're excited about.
Yeah.
So never stop learning. Number two, don't do something for a living. Do something that you're excited about.
Hmm.
That's mission critical. Everybody has their own limitations, but the more we expand our horizons, the better our chances of surviving, when we work with each other.
Hmm.
So I think read a lot. That's critical.
Hmm.
It was only in my second postdoc when I started to think. I've learned how to design drugs. I'm pretty good at it. When I have the right team, I'm able to identify chemicals which will modulate any protein. I'm sure there are stalwarts with millions of dollars in funding. Why aren't novel antimicrobials actually coming along? And one day I just sat there in the evening doing some literature surveys, and I came across this one paper from very prominent researchers in McGill. Um, and they were talking about the grand challenges that exist in antimicrobials. If I had never gone ahead and found that publication because I was interested to identify why we are not finding novel antimicrobials every day, I would never have learned what I've just told you today.
Hmm.
So, yeah, be curious, be interested. I think that's the biggest thing. And then enjoy what you're doing, obviously.
Right. Wow. And do you have any specific article, like reading material, that I can link in the description that you really like?
I could send you those.
Okay.
I really enjoy articles by um, Eric Brown. I really enjoy articles by Helen Skirskaya and Ribbenkoff from Oklahoma. There's one article in JMed Chem that I really love, which is by O'Shea and Mosier, which was published a long, long, long time ago.
Mhm.
Then there are people like Jerry Wright. Um, Definitely enjoy his work as well. Uh, I would recommend any and all of these researchers. They've done a magnificent job. They really have.
Okay, I will make sure to have a lot of reading material for our listeners. And I think with that, I'm going to end this episode. And Dr. Sarkar, this was an amazing episode. And thank you so much for giving us an insight. And also, I think I'm a little bit scared after this episode.
Well, if we never talk about the problems that we have, we'll
Yeah,
never find solutions. So I think this was good.
absolutely. Yes, absolutely. Well, again, thank you so much for your time. And listeners, I will catch you next week on another episode. And in meanwhile, if you have any suggestions, please email me at barka@nerdrxpodcast.com. And remember, it's good to be a nerd. Bye.
Thanks for having me. Bye bye.